Welcome, deep divers. Okay, picture this for a second. A human fetus, so tiny, so vulnerable, but it's alive and kicking, basically breathing underwater inside its mother.
It really is kind of mind-bending when you stop and think about it.
Yeah. It's pulling all the oxygen it needs, not from air, obviously, but straight from its mother's blood through, the placenta and the umbilical cord. How does that even work? It sounds almost, I don't know, magical.
It does feel a bit like magic, but it's pure, elegant biology, a real masterclass in physiological engineering.
So today we're diving deep into that, into animal physiology, right? Looking at how creatures, everything from a tiny water flea right up to a massive whale, how they get oxygen, how they move it around, and how they use it.
Exactly. And, crucially, how they deal with the waste product, carbon dioxide.
Yeah.
Because it's not just about lungs and gills, the obvious stuff.
Right. It's happening constantly, this molecular dance inside all of us and every animal out there.
So our mission really is to unpack all that, the concepts, the actual mechanisms, the systems animals use for this gas exchange.
We'll look at different strategies across different species, why these things evolved.
Yeah, the adaptive significance. And we'll even touch on, how scientists figured this stuff out. Some really clever experiments.
Get ready for some serious aha moments, folks. Because we're starting right there with that incredible beginning, how a fetus gets its oxygen.
Okay, so that fetus, its big challenge is getting oxygen from mom's blood, which, remember, has already dropped off some oxygen to her own tissues.
So it's not like it's getting fresh out of the lungs, fully loaded blood.
Not entirely, no. Her hemoglobin picks up O2 in her lungs, carries it down to the placenta. Then the fetus's hemoglobin has to basically pull that oxygen off the mother's hemoglobin.
Okay, but how, if her blood is already lower in oxygen, how does the baby's blood win that tug of war? Is it just diffusion or?
Here's the brilliant evolutionary twist. Fetal hemoglobin, the baby's version, actually has a higher affinity for oxygen than the mother's adult hemoglobin.
Higher affinity, so
Now,
it's stickier.
Exactly. It's chemically stickier for oxygen. Think of it like stronger magnets. The baby's hemoglobin literally pulls the oxygen molecules away from the mother's hemoglobin, even when the overall oxygen concentration, the partial pressure, is relatively low.
Wow. Okay, that's clever. So the baby can still get fully oxygenated even when mom's blood isn't saturated anymore.
Precisely. It's a fantastic piece of biological design. And that difference in affinity, that's all down to these molecules we call respiratory pigments.
Right, these are the key players, the molecular workhorses. So what's their basic job description?
Well, these respiratory pigments, like hemoglobin with its iron atom, or others we'll discuss, are basically proteins that can grab onto oxygen reversibly. That's the key. They pick it up in one place, lungs, gills, placenta, and let it go somewhere else, like in the muscles or brain.
Reversible binding. Got it.
And their most straightforward function, it's just dramatically boosting the amount of oxygen blood can carry. I mean, massively.
How much more?
Take human blood. With hemoglobin, it carries about 50 times more oxygen than if you just relied on oxygen dissolving in the watery part, the plasma.
50 times. Okay, that explains why we don't need hearts the size of, I don't know, washing machines.
Exactly. It hugely reduces the workload on the heart. But, they do more than just ferry oxygen around.
Oh. What else are they up to?
Well, hemoglobins especially are major buffers. They help keep the blood pH stable, which is critical. They're also involved in transporting CO₂, the waste gas.
Okay, buffering and CO₂ transport.
And then you have specialized versions, like myoglobin in your muscles. It helps pull oxygen into the muscle cells and stores a bit there, like a little emergency tank. There are even similar ones, neuroglobins, in nerve cells.
So myoglobin isn't just making muscle red, it's actually helping shuttle oxygen inside the cell.
It is. It facilitates diffusion. And the research is showing even more roles now. Some hemoglobins in muscle and nerve cells might even help regulate how the mitochondria, the cell's power plants, actually use oxygen, maybe by tinkering with things like nitric oxide.
Wow, okay. That's complex.
It is. And just a quick clarification, sometimes people say hemoglobin gets oxidized when it picks up oxygen, but that's not quite right. It gets oxygenated. Oxidation is a different chemical change, one that actually makes it lose its ability to bind oxygen reversibly. Oxygenation is just that temporary handshake, picking up the O2 molecule.
Oxygenated, not oxidized.
Right.
Got it. So we've talked a lot about hemoglobin, the red stuff. Is that nature's only solution? Or are there other colors in the oxygen-carrying paint box?
Oh, nature loves diversity here. Hemoglobins are definitely the most common. We have them, earthworms have them, even those little Daphnia, the water fleas.
Yeah.
But their structures can be wildly different.
How so?
Well, our hemoglobin is usually a 4-part molecule, and it's always packed inside red blood cells. But in an earthworm, Their hemoglobin is huge and just dissolved directly in their blood fluid, the plasma. Gives them that clear, wine-red blood.
And you mentioned Daphnia. They do something visible, right?
They're a fantastic example. If you put Daphnia in water with low oxygen, they actually start producing more hemoglobin. They literally turn bright red. You can see the adaptation happening.
That's amazing. What about other types? You mentioned other colors.
Right. So next up are the hemocyanins. You find these in arthropods like spiders and crabs and mollusks, think squids and octopuses. These use copper instead of iron.
Copper.
Yep, copper. No heme group like in hemoglobin. And they're dissolved in the plasma too. When they don't have oxygen, they're colorless. But when they pick up oxygen, they turn bright blue.
Ah, so that's where blue bloods comes from, like horseshoe crabs or octopuses.
That's exactly it. Then even more exotic, you've got chlorocruins, found in some marine worms. They're iron-based but have a slightly different structure. They look greenish in dilute solution.
Green blood.
Sort of. Greenish dilute but can look reddish concentrated. They call them green hemoglobins sometimes. And finally, hemerythrins. These are also iron-based but no heme, found in a few scattered invertebrate groups. They go from colorless to a sort of reddish violet when oxygenated.
Wow, a whole rainbow of respiratory pigments. You also said earlier they're like honorary enzymes. Can you explain that comparison?
Yeah, it's a useful analogy. Like enzymes, they have specific binding sites for oxygen in this case. They show high specificity. Binding oxygen causes a change in their shape, their conformation, and affinity, how tightly they bind oxygen, is a key property, just like substrate affinity for an enzyme.
But they don't change the oxygen itself.
Exactly. That's the crucial difference. Enzymes catalyze reactions, changing their substrate. Respiratory pigments just bind and release their ligand, oxygen, completely unchanged.
Okay. Now, something you mentioned with our hemoglobin having multiple parts, does that lead to interesting behaviors, like this cooperativity idea?
Yes, cooperativity is really important, especially for pigments like our blood hemoglobin, which has 4 oxygen binding sites. It basically means that binding oxygen at one site changes the affinity of the other sites on the same molecule.
So they talk to each other.
In a chemical sense, yes. For our hemoglobin, it's positive cooperativity. When one oxygen molecule binds, it makes it slightly easier for the second one to bind, and even easier for the third, and so on.
Like a team effort, getting the first one on board makes the rest join more easily.
Precisely. And this has a huge impact on how the pigment functions. It leads to a characteristic curve shape when you plot oxygen saturation against oxygen partial pressure.
Not a straight line, then.
No, not for cooperative pigments. You get a sigmoid curve, an S shape. Pigments with only one binding site, like myoglobin, or ones without cooperativity, they show a simpler hyperbolic curve.
And why is that S shape so important for hemoglobin?
That sigmoid shape means the curve is quite flat at high oxygen levels, like in the lungs, ensuring full loading. But crucially, it's very steep in the middle range of oxygen pressures, the range found in our tissues.
Steep.
Meaning that a relatively small drop in oxygen partial pressure as oxygen moves from blood into the tissues causes a large amount of oxygen to be released from the hemoglobin. It makes unloading incredibly efficient right where it's needed.
Ah, okay. That steepness is key for delivery. This brings us to really understanding that graph, the oxygen equilibrium curve.
Exactly. The oxygen equilibrium curve, the OEC, is fundamental. It maps out that relationship. Percentage of oxygenated binding sites versus the oxygen partial pressure. And a key metric we use to compare different pigments or conditions is P50.
P50, what's that stand for?
It's the partial pressure of oxygen at which the pigment is exactly 50% saturated. It's a measure of oxygen affinity. A higher P50 value means lower affinity. It takes more oxygen pressure to get it half full. We often talk about a shift to the right on the curve, meaning lower affinity, higher P50.
Lower affinity, higher P50, shift to the right.
Got it.
So let's apply this to us. How does human hemoglobin use this curve in our bodies?
Okay, think about the journey. Blood arrives at the lungs, low in oxygen. It encounters the air in the alveoli, which has a high oxygen partial pressure. Because of the OEC's shape, specifically that upper plateau.
The flat part at the top.
Right. Hemoglobin gets almost completely saturated, like 98, 99%, even if the lung oxygen levels fluctuate a bit. It ensures very efficient uptake. It's this amazing co-adaptation between our breathing system and our hemoglobin molecule.
Perfectly matched. Okay, so now the blood is loaded, heading out to the body tissues. What happens there?
Well, your tissues are constantly using oxygen for metabolism. So the oxygen partial pressure in the tissues is much lower than in the lungs. As the oxygenated blood flows past, Oxygen diffuses down its pressure gradient from the blood into the cells.
And as the blood loses oxygen, its partial pressure drops.
Exactly. And as the partial pressure in the blood falls, we move down the OEC, and hemoglobin starts releasing its bound oxygen. Now, at rest, your tissues might only cause the blood oxygen to drop enough to release about 25% of the oxygen it carried from the lungs.
Only a quarter, so there's a lot left over.
Yeah, quite a bit. That's the venous reserve. It's oxygen, still bound to hemoglobin in the blood returning to the heart. It's available if needed.
Ah, like during exercise. What happens then?
Exercise is where the system really shines, especially that steep part of the OEC. First, even during heavy exercise, the blood leaving the lungs usually remains almost fully saturated because of that plateau.
So uptake isn't usually the limiting factor?
Not typically in healthy individuals. The big change is in the tissues. Muscles working hard consume oxygen much faster, so the tissue partial pressure drops much lower. This pushes the blood further down that steep slope of the OEC.
And because it's steep.
Small drops in blood partial pressure lead to huge increases in the amount of oxygen unloaded. For instance, a drop in venous CO₂ of just 2 kilopascals, which is about 15 millimeters of mercury, can literally double the amount of oxygen released compared to rest.
Wow, so the S shape makes it incredibly responsive to tissue demand.
Totally. Plus, of course, blood flow increases dramatically to those working muscles, so they get more blood passing through, and they pull more oxygen out of each unit of blood. Under extreme exertion, muscles can extract nearly all the oxygen delivered to them.
It really feels like molecular design, doesn't it? The way human hemoglobin is structured to be nearly full in the lungs, have that safety plateau, but then have that steep unloading curve for the tissues.
Absolutely. It's not random. It's a consequence of its precise chemical structure, honed by evolution. And this system can be fine-tuned even further.
How so? What can change how sticky hemoglobin is for oxygen?
Several factors act as modulators. One really important one is the Bohr effect, named after Christian Bohr.
Okay, the Bohr effect. What does it involve?
It relates to pH and carbon dioxide. Basically, a decrease in pH, meaning it gets more acidic, or an increase in the partial pressure of CO₂, both cause hemoglobin's affinity for oxygen to decrease.
So more acid or more CO2 makes hemoglobin let go of oxygen more easily, shifts the curve to the right.
Exactly.
Yeah.
Shifts it right, increases the P50. And this happens because the hydrogen ions, H plus play, which cause acidity, and CO₂ molecules can bind to hemoglobin, but at different sites than where oxygen binds. They act as allosteric modulators, changing the protein's shape slightly.
And that's useful because
Think about active tissues, like exercising muscles. They produce lactic acid, lowering pH, and they produce lots of CO₂. Both these signals, right there in the tissue, trigger the Bohr effect, causing hemoglobin to unload more oxygen precisely where it's most needed.
ingenious. It enhances delivery automatically.
Yep. And conversely, in the lungs, CO₂ leaves the blood, pH rises slightly, and this increases hemoglobin's affinity, helping it load up efficiently. It's a dynamic, localized adjustment.
Okay, that's the Bohr effect. You also mentioned a root effect. Is that related?
It's related in that it involves pH and CO₂, but it's much more dramatic and much rarer. We mainly see it in fish, especially teleus fish.
More dramatic how?
With the root effect, lowering the pH or raising CO₂ doesn't just decrease the affinity, shift the curve, right? It also significantly reduces the maximum amount of oxygen the hemoglobin can carry, even if oxygen partial pressure is high.
It lowers the saturation ceiling.
Exactly. It effectively forces oxygen off the hemoglobin, preventing it from fully saturating even at high PO2.
Why would that be useful? Seems counterintuitive.
Fish use it for very specific things. Combined with a special network of blood vessels called a rate mirable, wonderful net, they use the root effect to generate incredibly high oxygen partial pressures. They essentially pump oxygen off hemoglobin into specific places, like the swim bladder to maintain buoyancy, or into the eye, particularly the retina, which needs a lot of oxygen but doesn't have many blood vessels.
Wow, okay, so it's like a biological oxygen concentrator. What about temperature? Does that play a role?
It does. Generally, for most animals, increasing temperature decreases oxygen affinity, shifts the curve right.
So warmer muscles during exercise would also get a bit more oxygen released.
Potentially, yes. It can work alongside the Bohr effect to enhance delivery. And this links to some amazing research, like the woolly mammoth study.
Oh yeah, you mentioned that, resurrecting ancient hemoglobin.
Incredible stuff. They used DNA from permafrost remains, about 43,000 years old, to synthesize mammoth hemoglobin in the lab, and they tested its properties.
And what did they find?
They found that mammoth hemoglobin was actually less sensitive to temperature changes, especially cold, compared to modern elephants. This suggests an adaptation to prevent oxygen unloading from being impaired in its cold feet and trunk. It needed to deliver oxygen efficiently, even when parts of its body were very cold.
That's evolutionary detective work at the molecular level. Amazing. Are there other modulators besides acid, CO2, and temperature?
Yes, there are organic molecules inside red blood cells that play a big role.
Hmm.
In mammals, the main one is called 2-pentyl-3-bisphosphoglycerate, or 2-pentyl-3-BPG for short.
2-pentyl-3-BPG, and what does it do?
It binds to hemoglobin and decreases its oxygen affinity, shifts the curve right.
Why have something that makes it harder to hold oxygen?
It helps fine-tune oxygen delivery. Think about someone who becomes anemic, meaning they have less hemoglobin overall. Their body often compensates by producing more 2-by-3-BPG. This lowers the oxygen affinity, meaning each hemoglobin molecule that is there unloads a larger fraction of its oxygen as it passes through the tissues. It helps maintain oxygen delivery despite lower carrying capacity.
Ah, a compensatory mechanism. Do other animals use PPT?
Birds use a different molecule called IPP, and fish often use ATP or GTP, the energy currency molecules, for the same purpose, modulating oxygen affinity. Even simple inorganic ions like chloride, calcium, or magnesium can sometimes influence pigment affinity in certain species. Blue crabs, for example, increase calcium in their blood in low oxygen to boost their hemocyanin's affinity.
It's a whole suite of controls. So these pigments are doing way more than just carrying oxygen.
Definitely. If we list it out, it's O2 transport, O2 storage, like myoglobin or in vertebrates and low O2 mud, blood pH buffering, CO2 transport, facilitating O2 diffusion into cells, even some enzymatic roles like managing nitric oxide, sometimes non-respiratory transport like carrying sulfide in some deep-sea vent animals, and potentially regulating mitochondrial activity. It's a surprisingly long list.
At least 8 functions. Okay, let's shift perspective a bit. How has evolution shaped these pigments across different species and environments?
Well, because they're right at that animal-environment interface, they're fantastic subjects for evolutionary studies, and we see clear patterns. One of those striking is that animals adapted to chronically low oxygen environments tend to evolve pigments with higher oxygen affinity.
Lower P50, shifted left.
Exactly. Their hemoglobin, or hemocyanin, is stickier, better at scavenging oxygen when it's scarce. We see this comparing fish like carp, which live in muddy, often oxygen-poor water, to mackerel from well-aerated open oceans. Carp have much higher affinity, HB.
And that's why goldfish survive in less than ideal tanks.
That's part of it. They're related to carp. You also see this trend in mammals that live underground, or species native to high altitudes. They often have higher affinity hemoglobin compared to their lowland relatives.
Interesting. Is there a pattern with body size?
In mammals, yes. Smaller mammals generally have higher weight-specific metabolic rates. They live life faster, metabolically speaking.
Like shrews versus elephants.
Right. And smaller mammals tend to have hemoglobin with lower oxygen affinity, a higher P50. This seems counterintuitive at first, but it means their hemoglobin unloads oxygen more readily in the tissues, helping to meet that higher metabolic demand per gram of tissue.
Easier unloading for higher metabolism. Makes sense. So that's evolutionary adaptation over generations. Can individual animals adjust during their lifetime? Acclimation?
Yes, absolutely. Animals can acclimate or acclimatize to changes in oxygen availability. The simplest way is often just changing the amount of pigment.
Like producing more red blood cells.
Exactly. Fish in low oxygen water often increase their red blood cell count, your hematocrit. Humans in high altitude initially do this too. But sometimes animals change the properties of their pigments.
How?
We already mentioned the Daphnia. When they move to hypoxic water, they don't just make more hemoglobin, they actually synthesize different types of hemoglobin molecules that inherently have a higher affinity for oxygen.
Wow, they switch isoforms.
They do. It's a qualitative change. Other examples, some fish reduce the levels of those organic modulators like ATP or GTP in their red cells, which effectively increases the hemoglobin's oxygen affinity. Those blue crabs we mentioned increasing calcium. That's another example of modifying affinity.
What about humans at high altitude? You said we increase red blood cells. Is that always the best strategy?
That's a fascinating area. For a long time, the increased red blood cell count, polycythemia, and increased 2,1,3-BPG, which lowers affinity, seen in lowlanders moving to altitude were considered the prime adaptations.
Maybe not.
Comparative studies show that many native high-altitude mammals and birds, llamas, Andean geese, bar-headed geese, often don't have unusually high red blood cell counts.
Oh.
In fact, too many red cells can make blood thick and viscous, harder to pump.
So the lowlander response might be wrong.
Some researchers suggest it might be a response evolved for dealing with anemia at low altitude, kind of misapplied at high altitude.
Hmm.
Many native highlanders actually show higher oxygen affinity, not lower, helping them load oxygen better in the thin air. It's complex and still debated.
Very interesting. And to cap off adaptation, we absolutely have to mention the ice fish again.
Ah, yes, the Antarctic icefish, Chenecthidae. Truly unique. They are the only vertebrates known to completely lack hemoglobin in their blood as adults.
No red blood cells, no hemoglobin, just clear blood. How is that even possible?
It's a combination of factors related to their extreme environment. They live in Antarctic waters, which are incredibly cold, close to freezing, and very stable. Cold water holds significantly more dissolved oxygen than warm water.
So there's just more oxygen physically dissolved in their plasma.
Yes, enough to meet their metabolic needs, which are also low because of the cold temperature. But they have other compensations too. They have much larger hearts, pump huge volumes of blood very rapidly, and have wider capillaries to reduce resistance to flow.
So they sacrificed the pigment, but compensated with massive circulatory changes, a complete physiological trade-off.
Exactly. A stunning example of adaptation to an extreme, stable environment.
Okay, we've spent a lot of time on oxygen. Let's switch gears to the other side of the coin, carbon dioxide. How do animals get rid of CO2, and how does this tie into acid-base balance?
Right, CO2 transport. It's not just the reverse of oxygen transport.
Yeah.
While a little bit of CO2 does dissolve directly in blood plasma, and some binds directly to hemoglobin.
But not at the oxygen binding site, right?
Correct. Binds to amino groups on the protein.
Uh-huh.
But the vast majority, typically 70% or more in mammals, is transported in the form of bicarbonate ions, HCO3.
Bicarbonate, how does that form?
CO2 reacts with water. CO2 plus H2O gives you carbonic acid, H2CO3, which then rapidly dissociates into a hydrogen ion, H plus Ana, and a bicarbonate ion, HCO3. Because this reaction produces H plus anima, CO2 essentially acts like a gaseous acid.
So generating bicarbonate makes the blood more acidic.
It would, except that the blood has powerful buffers that soak up most of those H plus ions. And the most important buffer in vertebrate blood is
Hemoglobin again.
hemoglobin again. By buffering the H plus ana, hemoglobin allows much more CO2 to be converted into bicarbonate and carried in the blood without drastically changing the pH.
So hemoglobin is crucial for CO2 transport capacity too. Is there a CO2 curve like the OEC?
Yes, the carbon dioxide equilibrium curve, COEC. It plots the total CO2 concentration in the blood against the partial pressure of CO2. Its shape differs between air breathers and water breathers because they operate at different typical CO2 levels.
And is there an interaction between oxygen and CO2 transport, like the Bohr effect relating CO2 pH to oxygen affinity?
Absolutely. It's called the Haldane effect.
Yeah.
It's essentially the flip side of the Bohr effect.
Okay, Haldane effect. What does it state?
It states that deoxygenated hemoglobin is better at picking up CO2, both directly binding CO2 and buffering H plus from bicarbonate formation, than oxygenated hemoglobin. So as blood releases oxygen in the tissues, its ability to take up CO2 increases.
So oxygen release helps CO2 uptake.
Exactly. And conversely, when blood gets oxygenated in the lungs or gills, its CO2 carrying capacity decreases, which helps unload CO2. Deoxygenated hemoglobin is simply a better proton acceptor, a better buffer.
Wow. So the Bohr effect, CO2 pH affects O2 binding, and the Haldane effect, O2 affects CO2H plus binding, work together beautifully. Hemoglobin facilitates both gas exchanges simultaneously just by cycling between oxygenated and deoxygenated states.
It's an incredibly elegant system, a symphony, as you said earlier.
Yeah.
Now, that reaction of CO2 with water to form carbonic acid is actually pretty slow on its own.
Slow, but gas exchange needs to be fast.
Right. So there's an enzyme that speeds it up dramatically. Carbonic anhydrase, or CA, is one of the fastest enzymes known. And crucially, in vertebrates, it's mainly located inside the red blood cells.
Inside the red cells, but most CO2 is carried as bicarbonate in the plasma.
Correct. So CO2 diffuses into the red cell. Carbonic anhydrase rapidly converts it to H+, buffered by HP, and bicarbonate. Then the bicarbonate needs to get out into the plasma
How does it cross the cell membrane?
through a specific protein transporter. And as the negatively charged bicarbonate ions leave the red cell, negative chloride ions, CHULO, move into the red cell to maintain electrical balance. This is called the chloride shift or hamburger shift.
The chloride shift. Okay, lots of moving parts. This all ties into maintaining the body's overall acid-base balance, right? Why is pH so tightly controlled?
Because proteins, especially enzymes, are incredibly sensitive to pH. Their structure and function depend on the pattern of electrical charges on their amino acids. Even small shifts in pH can alter these charges and severely impair protein function. For us, normal arterial blood pH is about 7.4. Deviate much from that, and things go wrong very quickly. It can be life-threatening.
And this balance can be disrupted.
Yes. We talk about acidosis, pH too low, too acidic, and alkalosis, pH too high, too alkaline. These can be either respiratory or metabolic.
Respiratory versus metabolic.
Respiratory disturbances are caused by problems with CO₂ elimination. If you hypoventilate, don't breathe enough, CO₂ builds up, forms acid, causing respiratory acidosis. If you hyperventilate, breathe too much, you blow off too much CO₂, leading to respiratory alkalosis.
Okay. And metabolic.
Metabolic disturbances involve changes in bicarbonate levels or the accumulation of other acids or bases. For example, intense exercise can produce lactic acid, causing metabolic acidosis. Kidney failure can also disrupt acid-base balance. The body usually tries to compensate, often by adjusting breathing.
So if you have metabolic acidosis, you might start breathing faster to lower CO₂ and counteract the acid.
Exactly, that's respiratory compensation. And finally, this whole issue of CO₂ and pH has huge relevance today on a global scale.
You mean ocean acidification?
Precisely. All the extra CO₂ we're putting into the atmosphere, a lot of it dissolves in the oceans. It reacts with water, forms carbonic acid, and lowers the ocean's pH. We've already seen about a 0.1 pH unit drop, which is a significant increase in acidity.
And that's bad for marine life.
Especially for organisms that build shells or skeletons out of calcium carbonate corals, shellfish, plankton. Increased acidity makes it harder for them to build their structures and can even start dissolving existing ones. It's a major threat to marine ecosystems, a direct physiological consequence of global CO₂ changes.
Wow. From the fetus's first breath to global ocean chemistry, it's all connected through these fundamental processes of gas transport and acid-base balance.
It really is. We've gone from metal atoms and proteins all the way to whole animal adaptations in extreme places like Antarctica or high mountains. It underlies so much of physiology.
So as you listen, think about your own body right now. Every breath you take involves this incredible silent ballet of molecules, hemoglobin loading oxygen, releasing it, picking up CO₂, buffering pH. It's happening constantly.
Yeah, maybe the next time you see an animal, whether it's your pet or something in the wild, just take a moment to appreciate the complex chemistry keeping it alive, that constant exchange of gases.
It definitely makes you think. We've seen this intricate molecular design, especially in hemoglobin, shaped over millions of years. A final thought, maybe. In a world that's changing rapidly now, how much more can these physiological systems adapt? Can evolution keep pace?
That is the big question, isn't it? And a critical one for the future. This deep dive is just scratching the surface of animal physiology, of course. There's so much more complexity hidden in seemingly simple functions. Keep asking questions. And thanks, everyone, for joining us.